My name is Becky Suker, and I'm the Senior Manager of Living Collections here at Missouri Botanical Garden in the Horticulture Division. And I'm here to tell you that living collections are really the core purpose of a botanical garden. By definition, botanical gardens are institutions that hold documented collections of living plants for the purposes of conservation, scientific research, education, and display. It's these collections that are identified, recorded, labeled, and curated that are really valuable for scientific research and conservation. So some of the things we do include targeting species for the living collection. We're really interested in adding species to the collection that we don't already have, that are new and different, that are threatened or endangered in some way, that are beautiful or unique or iconic, or help us tell a story and educate the public in some way. Once we target those collections, we work to acquire them. So we plan expeditions around the world and around the country, and that involves a lot of trying to figure out where the plants are, gathering data, figuring out when is the best time to collect a particular plant, when it's in seed. Then we have to plan all of the logistics around collecting that, like renting cars and renting places to stay and what we're going to do on what day. And then gathering supplies, applying for permits. Once we are in the field and collecting plant material, we try to record that plant material at the highest level possible. So we're recording everything we see and know and can learn about that plant in the field. And we do that in our collecting books, and we record the name and the date of the plant was collected, the plant material that was collected, whether it's seed or cuttings or divisions of the plant, the latitude, the longitude, the habitat, the sun exposure, the soil. All of these things help us understand more about that plant and enable us to grow it in cultivation. Once we bring that material back to the garden, we record it, we accession it. And accessioning is a process of assigning an ID number to a plant. So we're recording that name and that number and then all of that information that we collected in the field. That we then produce labels and zinc. Some of the labels you see are for the public to be able to know what they're looking at, but others are called accession tags that allow us to tie all of that data that's in the database to that exact plant. And that tag is really one of the most important things that we can keep that connection between the data and the plant. Once we're growing that plant, we also facilitate the recording of other information into the database. We record photographs, we record all of the permits and agreements and other documents associated with that collection. And then we record propagation protocols and the successes and the failures that we have along the way. In building the living collection, one of the goals is to build as much species diversity as possible. This means that some of our collections are kind of like living libraries that allow our scientists to study a wide range of species in a very small space. Additionally, these plants have all of that documentation and source information associated to them. I'm here in the Orchid range, which is a collection of plants in the family Oraceae. This is a great example of a living collection, and we have living collections for many purposes, and there's two main purposes for this collection. One is just that the place where these plants were collected from in the tropics, mostly in Central and South America, their habitat is disappearing really fast. And so by bringing these plants into living collections, we're acting as a safety net against their potential extinction. Additionally, these plants are really hard to identify by just their leaves. As you can see, a lot of these plants look very similar. And one of the unique identifying characteristics of plants in general is their flower. And so when you're in the forest or the glade or the prairie or wherever you are, you're not necessarily there when it's in flower. And so by bringing these plants back into a living collection, we can cultivate them, observe their flowers, and then use that information to classify. If maybe it's an unnamed species, we can name the plant, understand the other plants that it's related to, and be able to further study that plant in cultivation. So this greenhouse that we're in now contains some of our plant collections from the island of Mauritius. Mauritius is a small island in the Indian Ocean just east of Madagascar. It's about 30 miles wide by about 40 miles long, but there's a population of about 1.2 million people on that island. So as you might expect, the native forest cover has been reduced to about 2% of its original size. Mauritius is also special because it has an incredibly diverse flora. There's 700 species native to Mauritius. Half of those are endemic, meaning they occur nowhere else in the world. Unfortunately, about 90% of those species are critically threatened with extinction, and 60 species have already gone extinct. So we're working with our colleagues in Mauritius to help protect the diversity and the flora of Mauritius. We are working with stakeholders from the National Park Service, from the forestries, and from the Mauritius Wildlife Foundation to locate, map, and learn how to propagate some of these species so that they can be reintroduced to the wild. Because the island is so populated, the plants have really only occur in places where people don't. So this is on tops of mountains, on sides of cliffs, and it's really hard to get a handle on how many plants are left because they're really hard to access. And so one of the things we did on our last trip was to actually use drone technology to help get a handle and to see if we could figure out, understand more about these populations. And so we were able to actually map in 3D a whole area called PEDS 500. It's a waterfall, and it's a pretty well-known spot, and it's the most well-known spot for where this species occurs. And so because it was on the sheer cliff face, people only could guess at about how many plants might be there. But we were able to 3D map the entire area and take thousands of photographs, which has taken us months to stitch together and really look at, and we've been able to count at least about 200 plants. And so that's a really exciting thing to prove this concept of being able to use a drone technology to assess these populations. The species that we were actually looking for on that waterfall was Anesicodon mauritianus, which we have growing here. This is the Mauritian bloody bellflower, and it's called that because the nectar is actually a blood red color, which is really rare in the plant kingdom, and it's actually pollinated by geckos. Whenever we're on a trip which is planned around a target species, we also collect opportunistically. So that means if there are other species that are producing seed that maybe we weren't planning on, we might collect seed from those as well. And so we have some of those in this greenhouse. One of those is Cyadia polycina, which is in flower right now, and that's also a critically endangered species. And the plan for some of these is to show that actually some of them are already located in our Climatron Conservatory, where we're learning how to grow them in cultivation, but also sharing these spectacular plants with the public and the story of plant conservation and of the flora of Mauritius. So another plant we have in this greenhouse is Bricamium cygnus. This is actually endemic to Hawaii. It's called cabbage on a stick, and you can see why. And back in 1994, there were about 5 populations of this plant known to exist in the wild. It grows on cliff faces in Hawaii. And unfortunately, today, it's thought to potentially be extinct in the wild. The last known recorded location was documented in 2014. So This is a really sad and scary thing because this plant has gone extinct on my watch. Since I've been working here at the Botanical Garden, to watch a plant go extinct in the wild is a pretty scary thing. But on the good side of things, on the positive note, this plant is well represented in living collections in botanical gardens. And those collections are really well documented, so we know where those plants were collected. Additionally, there's been a lot of genetic work done with this species. And we know now that what is held in botanical gardens represents pretty much the diversity, the genetic diversity that existed in the wild. And so the next step, and this is ongoing research that is happening right now, is trying to determine how to cross these species so that we can maximize genetic diversity. Because genetic diversity is really, really important when we're bringing these plants back into the wild. That's going to help them be able to adapt and evolve and deal with the pressures of the wild and nature. When this plant is in flower, we've shared pollen with other botanical gardens, and so they're conducting controlled crosses with individuals of known provenance, where they came from, and this is allowing us to be able to maximize the genetic diversity in the resulting seed, and that those seeds can then be used to grow more plants that could potentially be used to be reintroduced into the wild. So as you can see, we're building collaborations all over the world, from places like Africa, the Caucasus region, Central Asia, and even here in the United States, to help find, record, and grow plants of conservation concern, and also to advance conservation horticultural skills and to build capacity for living collections development and management. One of our most recent projects is here in Missouri. It's with the Eastern Prairie Fringed Orchid, Platanthera leucophea, and we're working with the Missouri Department of Conservation and the U.S. Fish and Wildlife Service to help learn more about how to grow this species outside of its native habitat. So the project entails visiting the site where the Eastern Prairie Fringed Orchid occurs in northern Missouri. It's threatened with extinction, so it's a really small site, and the number of actual plants varies from year to year. And we went during the flowering stage and actually hand pollinated the flowers. Because this population is so small and fragmented, the seed set isn't very reliable, so by hand pollinating the flowers, we can ensure a better seed set. We can also ensure that the flowers are getting cross-pollinated. And so by hand pollinating, we were accomplishing a few things and just making sure that the project was a success. A few weeks later, we went to collect the immature seed, and we were excited to find out that our pollination efforts were successful, and we were able to collect immature seed from all 10 plants that we cross-pollinated. And those are currently in our micropropagation lab, and just found out today that we are seeing some germination on those early seed, on the immature seed. So that's really exciting. Our team also went back a third time to collect mature seed. We're also taking seed from that collection and storing them in our seed bank. So where we're freezing the seed so that we can actually use it further down the line if we want to, for a potential research or conservation project. The goal is to eventually get the plant out in the garden to share with our visitors so that you can see this beautiful plant. This particular prairie is a wet prairie, and finding a place where we can grow it here at the garden will be a trial and error. We often have plants that we're bringing into the collection where we might not have the perfect situation for it, and so we try it in lots of different places, and we try different soils and different shade situations and different watering techniques, and record our successes and failures and how well the plant is doing. And so that's all part of conservation horticulture and how we are contributing to conserving species outside of their native habitat. So thank you for joining me on this little peek behind the scenes of how we target species for the living collection, what we choose to add to the living collection, how we record and maintain our records, and how we propagate and grow plants. As you can tell, our collections are constantly growing and changing, so we look forward to you coming and visiting and seeing some of the new things that we're adding. Thanks again.